So I've got three themes here. Are you all well installed? Is everything okay? Yeah. So the first theme that noted and that has come up a few times is about the evolution of gene order and order also inside a gene with the domains. Then the second theme that we're going to discuss later is about isoform evolution or transcript annotations that was discussed in Aida's keynote, but also that came back also in some of the talks, and where I think we have a real challenge in front of us. And then finally, we're going to tackle the theme of large-scale phylogenetics, which is something interesting both for cancer phylogenies, for pathogen phylogenies, but also all of the genomes. And I can see a few people nodding their heads in the audience, so you can look forward to that. So, but let's start first maybe with with synteny, the order, the conservation or not of the orders of genes, as well as constraints within the domains. And this is a discipline, particularly at ISMB and in this community, that is almost like classic and that has been looked at different levels. And it's clearly something really important. I mean, I thought we had some interesting and forceful evidence also of the constraints that are there. But it strikes to me that the algorithms that have been developed have not really yet reached the mainstream. And most alignment approaches and genome analysis are not really tackling this problem, let's say, in an algorithmic manner. It's usually quite ad hoc. And so I'm wondering whether you have some thoughts on this and what you think might be still the stumbling block there, and what we might need to do there. So I don't know who wants to start.
Would you like to start, since your lab has had?
I have all sorts of cranky opinions about synteny. So let's see, where do we start? One issue with understanding spatial organization repeats, and this happened to come up at lunch, is the problem of gene conversion, and that's something that I think is underappreciated. But Giltai has a paper on it, so at least one person appreciates it. So there were these within proteins and domain architectures, there are lots of very compelling examples of strings of repeated domains. So for example, in zinc finger proteins, the number of copies of the zinc finger are crucial to how it's going to bind the DNA. And there's a great temptation to try to analyze these using sequence comparisons. And a problem that frequently arises is that the DNA repair mechanisms, you'll get unequal alignment of the copies. And then the DNA repair mechanism will use a copy of an adjacent domain to correct errors in a neighboring domain. And so you're constantly overwriting the phylogenetic information that is normally embedded in sequences. And I suspect that there are a lot of errors that there are a lot of papers that report results which are in fact incorrect because accounting for gene conversion is not normally considered. And there are methods out there. They're hard to find. You have to find somebody who's done this and then dig through their method section. But I also think you can test to see if gene conversions occurred. And that protects you against, incorrectly analyzing your data. If it has occurred, I'm not sure what you can do about that. Whether you're then getting into some twilight zone where you just have no information. So short of doing something really clever with structures, perhaps. So that's one thought about sort of spatial questions. Another observation would be that we are only recently seeing these emerging because you have to have good quality assemblies in order to really know what the spatial organization is. And so, now that we're getting long read sequencing, we can begin to think about that. And in fact, a family with repeats that was discussed at lunch, there's a great deal of polymorphism within, different strains in the same bacterial species in the number of copies of these repeats. And there's a theory about sort of it allows bacteria to reach out to the host at different distances depending on the length of the repeats. But long-read sequencing was crucial to being able to do that analysis at all.
Well,
So
thank you very much, Danny. How about then the issue of synteny? Perhaps, Aida, do you have any thought on this? I mean, we've seen some methods that can detect duplications in terms of blocks. I mean, but these are also not really used very much then in practice. I mean, where the analysis in comparative genomics just consider one gene at a time. What's missing, you think?
Okay, thank you it's an interesting question as well. And I will continue with what Danny was saying actually. So yes, so the algorithms at the moment are not integrating all the events that we know that are implicated in the evolution of these genes, of these centenies of genes, or in the evolution of domains. But I think this is because of the complexity that they bring to the problems, and so to the algorithm that we can find to solve those problems. This is one thing. We should go towards integrating more models of evolution, accounting for the complexity actually of the mechanism. But there is another problem as well, which is that we don't know exactly all the evolutionary events that are actually happening to explain the evolution, whether it's for centenies of genes or it is for the domain architecture. And I think one over direction that can also help is development of, since we don't have all the data yet, it's the development of simulators, tools to simulate the evolution. And with such tool, actually, which is interesting, we have seen an example of presentation this morning about evolution of domain architecture. What is nice about this kind of tool is that we are able to test some hypotheses. We are able to even go toward finding and better understanding, actually, the evolutionary histories of this set of genes, centenic genes, or domain architecture. And this is another direction I think that should be further digged.
If I could sort of interject, I think there's the theme that's come up, a couple of times here is one of data and quality, and obviously long read sequencing is becoming a game changer. But in terms of large scale synteny, we face a couple of things. One is the taxon sampling for good long read assemblies, especially in vertebrates. And I'm sort of putting prokaryotes, which are more advanced, a little to the side. So maybe they're a good test ground. But if you think about vertebrates, what we have is limited taxon sampling of these very high quality assemblies. We have heterogeneous qualities even within the very good assemblies. Some are very, very good. Maybe they are including, bio-nano and nanopore and PacBio data. Others, maybe less so, but still good. So I think coming up with parameters, at least for the next several years, for estimation and simulation, is going to be really fundamentally challenged by the absence of data to estimate those parameters.
Thank you. I'm wondering also, I mean, at the moment, but perhaps the assembly problem is going to be largely solved with long reads. I mean, still, particularly for the for the groups who cannot afford, the like really high quality long read assemblies. I mean, perhaps this community could also help by the fact that all the genomes are related, and then this order is also somewhat conserved. And so it also strikes to me, particularly as we sequence denser and denser and very closely related species, that we perform this assembly independently for each of these animals. So maybe I'm broadening this question also perhaps also to the audience, if some of you are familiar with tools that are trying then to not only exploit maybe the outputs of these assemblers to study the evolution of the positions of genes or domains within genes, but perhaps also exploit these relationships to improve assemblies.
And I should emphasize that we really had envisioned this as involving everybody. So if you have ideas, be running up to the microphone or raising your hand. But if I can just very quickly point out that, Christoph's point about using available high quality assemblies to improve the assemblies of close relatives is conceptually quite similar to the use of variant calls in cancer. Our very first talk, which was, I think, very clever, to improve calls of the mutations. So I think there's something where the community could help.
Thank you for that very good reference. There's also another kind of emerging data that maybe none of us are real experts on this panel, but has been represented in this session, and it is very exciting, is all the cancer data, and it's understood that structural variation can have a big impact in many cancers. And it seems to me that that's also a case where the different communities have worked quite separately, but where there could be some interesting connections. And so the same way we've had, like in the past few years, a number of talks that bring together the cancer and the classical, let's say, evolutionary community together for the trees. Perhaps we'll see something similar in terms of structural variation. Dannie, you have some thoughts?
Well, I was on a more perhaps philosophical or higher level. It's really interesting to think about to what extent does evolution in tumors represent sort of a speeded up version of evolution that has occurred over in genes and species, and can we take advantage of the kinds of changes we see in tumors to set parameters, for example, or how do we try to figure out whether or not those are different processes and we shouldn't be using them as estimates? So I think that's a really provocative question. I don't have good ideas about how to get started with it.
I mean, the other point is just the sheer quantity of data and how human is becoming the model organism for actually a lot of biological processes. I mean, it started off the other way around, that we were using other species to understand better the human. And now it seems that it's all these human genomic data, be it like from healthy tissues or cancerous tissue, that are teaching us something about these very fundamental processes. I mean, isn't it funny? So I don't know if there are other comments, but please, or questions on this topic, feel free to still, come to the microphone. Yeah, guilty. So thank you.
Yeah, I have 2 points. If we want to infer like a gene tree or phylogenetic tree of a species, then like having good quality of assemblies could help to improve it. But my question is like, what about if we want to infer like gene tree, then we need a gene annotation. But then but do we have like good annotation tools so then if you have some suggestions, there are multiple exons like in a gene, then it's quite complicated. That gene itself is quite complicated. And so in my experience, the annotation tools quite doesn't work well. And so, but you have a experience, then you have if you have any suggestions, then yeah, that's the first. And another is like for cancer genomes, do we get like assembly like for cancer sequence, cancer genome sequence? So we have that kind of technologies, and yeah, so that's yeah.
Yeah, so I'll just mention perhaps the second point is your first point is going to be a perfect transition to the second theme. But so I think for your second point, there are single cell cancer data set that are being published. They are low coverage, and usually the information that is obtained is still just very kind of local order. I mean, because of I mean, maybe using just paired read or it may identifying short structural variants. So you don't usually have like a full genome assembly, but nevertheless, I think I mean you don't need, I think, to be really visionary to imagine that there's going to be more sequencing, and this is going to be resolved. And actually, I think even with just something fragmented, you could get some local synteny and start studying a lot of things the same way we've done it actually with lots of genomes across the tree of life classically.
Yes.
So yeah, I wanted to add also about the reconstruction of, for instance, cancer phylogenies. The question is actually whether we are at the moment using the right models and algorithms to reconstruct them, but because for now we are extending what we knew before when we were reconstructing the evolution of genomes. If I take, for instance, the whole theory of gene rearrangement that is actually being used as it is for the reconstruction of cancer phylogenies, we see that the events are more complex. Like, for instance, we will not cut at only two places and then replace the position to have a rearrangement, but we have kind of events that happen that look like we are cutting at many different places at the same time and regluing the genome, passing the genome in a different form. So are we using the correct model, or should we use models that are completely different for cancer? So that's the question.
Okay, thank you, Aida. So I think we can move with your first point actually to our second theme, which is exactly that, which is that we might solve the problem of assembly, but the problem of annotation really seems a real challenge, and there does not seem to be much progress. We've I think heard like a very forceful illustration of this as part of Aida's talk. I think those of us working on comparison across different genomes have all been limited in one way or another. It's also interesting to see the community that works on transcriptomics is also usually mapping at the level of genes, or maybe at best exons, because of this limitation quite often. And so there I want to ask some really open-ended question, which is, I think we need some more collaboration across areas. And I wonder what sort of collaboration, with which other causes and other areas of computational biology could this community collaborate to really tackle this challenge as one of I think one of the possibly the grand challenges of bioinformatics, which is how do we solve the problem of annotation?
Where annotation?
I mean, identifying the genes, identifying the transcripts, the
Gene models.
The gene models, yes.
can start. Okay. Yeah, that's another very interesting question. And actually, I will, I don't have an answer, but I will go even further. So, yeah, you were talking about working with people that work on transcriptomics. But actually, we have the two opposite problems. So the problem that we have incomplete data, because right now the annotation are not complete for all species, but we know with the development of the RNA-seq technologies, now we are able to sequence the transcriptome and to know more about what are the transcripts that are produced by genes. So the opposite problem is that we may end up with too much data. And I will explain myself. Too much data because, for instance, we've been discussing, it was yesterday or the day before, with someone who had this sequencing at the level of molecules, and who ended up with, for some genes, several transcripts, so many transcripts, more than 100, when the canonical and the few ones that were in the database were maybe 5 or 6. And the question were actually which one of these transcripts are the real ones that are really expressed and are useful to be regarded in phylogenetic reconstruction. So yeah, it is true that we need a group effort with many different thematics working together in order to get not only complete but error-free annotation. But then there are other questions as well if we mix the experiments with the theoretical parts.
Thank you. This was Anna Conesa, I think,
Yes.
who was with whom you discussed. Yeah. So and so in the back of my mind, I'm thinking where evolution can help. If you have really a wealth of transcripts, the deeper you sequence across different species, is that the things that are really important are going to be selected and are therefore going to appear across different species. And all the alternative splicing, one might think, might be evolving faster than, say, folds of proteins. But nevertheless, as we get more and more closely related species, surely not every species has reinvented the regulation of like the different differentiation across organs, etc. That is possibly also partly regulated through alternative splicing. So I don't know if there are any thoughts on this, but to me it seems that more than any other topic, that's really a place where evolution and an integrative approach across the tree of life could really provide some help.
Well, I think you have a circular situation where we don't really know how fast those things evolve. So on the one hand, we can learn more about how fast do changes in splice forms and how they're regulated occur across evolution. And as we start learning that, we can also try to use it to get better annotation. And the trick is not to fall into this hole where we get the wrong annotations and learn from that. But it's also a great opportunity to learn those things.
Yeah, so I was going to sort of follow on with Danny's thought, which is obviously there is this potential of propagating problems. Think of all the annotations that have propagated that are weird just due to blast. We annotate, we find X blasts to Y, therefore we call X, which is in an organism for which we don't have functional data, a Y, and Y turns out to have been kind of flaky in the first place. We ideally would not want to repeat that problem. And I think one of the challenges we have with deeper and deeper RNA-seq and deeper ISO-seq and whatever other technologies come along is how much of this is essentially errors in the transcription splicing polyadenylation machinery versus forms that are actually biologically significant at a reasonable level. And, you know, what can we do to separate sort of the signal from the noise? And evolution may be exactly what we need to separate that.
I think,
Any thoughts from the audience? Yeah, or
no.
well,
like,
Danny.
Audience?
Meanwhile, Danny.
Well, so you were describing contamination in databases. I think there's another problem, which is sort of conceptual contamination. We have an understanding of how biology works, and then we build tools that exploit that. So you have a colleague, Aida, who there is a general view that genes must be at least 100 nucleotides long to encode a reasonable protein. So nobody looked for genes that were much shorter. And then when Xavier Huku discovered all of these peptides that are much shorter than that, and that are often within genes, but in a different, reading frame. So it's just one example of, once we have a conception of how biology works, we're likely to rediscover, it's confirmation bias. And that's something we have to be careful about.
Lars.
Yeah, I dare myself up here. I am worried about something similar to what you're saying, and that is sort of a conformance or conservatives if we start trusting on evolution as reproducing things. So, I mean, many times when we're looking at closely related species or two species, etc., we're looking for the difference really. And if we have done a lot of our annotations by looking at evolutionary conserved features, then we might not simply find those distinguishing features or underestimate them severely.
So that's true. That's true. So we are inherently biased towards a thing that we can compare, that are conserved. But I must say, I mean, so at least in my personal experience, I mean, quite often when we see differences, it's I mean, there are much fewer differences once you've really cleaned out all of the artifacts and so on. So, we have so many functions that are conserved across billions of years. I mean, at many levels, I mean, we are very similar to, I mean, we like to think of ourselves as really special, but actually we're extremely similar to lots of other primates and mammals and vertebrates in many ways. And so I think if we could do a good job, I think, identifying the aspects that are in common, then maybe we can then tackle the new stuff. And I mean, and we could, and I think the point about the noise and our uncertainty is really valid. But we could start more modest and just thinking, okay, can we at least identify the splicing variant that is best conserved across evolution, which is something that ridiculously we don't even do today. Most of the time we just assume that is the longest one that we are going to use to compare across different species.
Well, and this is a community of computational biologists. Can we come up with really robust statistical models that would help us try to distinguish between noise and differences? So that it seems like there are interesting problems there for people to work on.
I think that this is a question for every, perhaps, where, maybe in a few years you are going to tell us about the difference between like a selected alternative
Next week,
splicing variant.
we can't wait a few years. Get going.
Right now. And also, I just wanted to add about the aspect of the conservative side. Actually, I think we are talking about model-based and model-free approaches. Okay, it is true that right now we use model-based approaches, we are not using them enough in order to complete the annotation. And what Christophe is saying, actually, we could use them to go further and complete the annotation and even correct some errors that are already in the databases. And we should also work on model-free approaches to discover new gene models or new events, like I was talking about before. So both of the directions should be, I think, digged.
Thank you, Aida. So I'm conscious of time, and I do think we should move to our third topic, which is about the very large scale phylogenetics. And so, as I mentioned very briefly in the introduction, this is coming from all sort of direction for all sort of from all kinds of data, and we see these enormous sequencing efforts like around the Earth biogenome umbrella, with the ambition to sequence more than 1 million eukaryotes across the tree of life within the next decades. But then just like within a few years now, we've had all these COVID sequences. There are now tens of millions of sequences that are available, which have challenged not only the existing pipeline, but also even our conception of phylogenies, with trees that have mostly consist of branches of 0 length, and where we're really somehow still really concerned about resolving the fine detail of the phylogeny, and then measuring support for these extremely short branches. We've heard quite a few interesting talks, I think, to tackle these problems. We've heard also talks from cancer phylogenies, I think, which also need to tackle very large data set. We heard about the sort of assumptions, and well, in some cases even algorithmic improvements with some exact methods that enable to compute some of these trees more exactly. But I want to ask very broadly now, if we project ourselves and we're in this world with essentially where the data is unlimited, for instance, does it still make sense for us to think of our different methods in terms of their statistical efficiency when we're comparing, for instance, different tree building method? Is it better perhaps to have a kind of simpler method, but that are more scalable and that can integrate more data? Or on the contrary, that's a really bad idea. We end up with all of these very short branches, and we should rather subsample. As we've heard, we could subsample columns, as the lasso talk yesterday suggested, or perhaps subsample in a different way. So I'm sure that our panelists and the audience will have some thoughts on this.
I'll just start to follow up on something that's not really my core area, which is the COVID sequencing. And I'd sort of like to get a feeling from the audience as to what you think is going to be most valuable way moving forward. Obviously, COVID is going to be with us for a while, and it's becoming endemic. What's going to be more valuable? Continued sequencing of more COVID isolates, sequencing out of, say, sewage effluents or something to just monitor communities, or potentially going out and doing surveys in nature, trying to look for other coronaviruses, or other things. But certainly coronaviruses have emerged how many times in recent history, with SARS and MERS, and well, now the bad luck of SARS-CoV-2. So what do people think of those three strategies? What would be most interesting? Or the panel?
Well, I'm sorry, I am not going to answer the question, but I just wanted to say that for the people that are online, you can participate. And actually, we were not looking at the answers, but before we had the contribution from Nadia and Janani. So Nadia was saying, I guess it was at the beginning when we were talking about the partial data. We are still working on that. The algorithm at the moment are partial. We need to integrate a lot of information at the same time. Yes, that's two gene order, gene trees, many types of event repeats. Janani was saying, excellent point. Evolution and comparative genomics might be perfectly positioned to work alongside MLS-CSB and NetBio causes for large-scale data imputation to address the missing data issue, and function COSI for annotation. That might be a small start. Cross-COSI collaboration sound like the way to go. And I'm wondering, for those that are online, are you able to participate with your microphones, maybe? No, they are not. Okay, so you can use the chat if you want to contribute.
So we have Thomas in the room, and I'm sure that Thomas might have some thoughts on COVID sequences. It's been a topic of keen interest.
Yeah, I'm not really an expert in evolutionary genetics, but I just want to mention that in addition to the two sources of information, namely sequencing infected patients and sequencing putative natural reservoirs, there is a third repository that we had a talk on just this morning in the COVID session, namely the SRA archive, where the Ceratus project basically used that archive, which is very voluminous and which is, as according to the speaker there, Artem Babayan, who is one of the activists in this consortium, is underappreciated and basically used as a dump. So that archive has a lot of signals, and they have a technology in which they try to unearth these, and they have very interesting questions. As a matter of fact, so this archive, there's a recent publication that where they claim to have 130,000 new viruses that they found in this archive, which is tenfold the universe of all the viruses that we know, but they claim it's still like, 1 per mil of what's out there. The initiation to that project was COVID. They were thinking about what are we going to do about future pandemics and where can we find viruses before they find us. And so they have, again, I've just heard this talk, so he would be much better to talk here. But so they have also signals on COVID, and he asked the question whether COVID possibly is a segmented virus. If that were the case, that would be, an issue, because segmented viruses can evolve in much more rapid ways. So I just want to mention that looking at all the stuff that we have sequenced under some context, some very narrow contexts that we're not thinking about being valuable in a broad context, and we have spent billions of dollars sequencing these things, maybe another treasure trove here.
Well, thank you, Thomas. That's for our points very well taken. This reminds me also a little bit about the Terra Ocean expedition that also characterized about 5,000 new viruses, of which only about 17 or 18 had been in the part of the international classification, which just shows like the vast size of our lack of knowledge.
So I just want to add one thing. So that one question I asked about this, he did this about viruses, but of course biodiversity is much broader. So and your radar is much broader. You're not virus evolution, you're evolution. So would that technology also help you? Would, raking the SRA help you uncovering evolutionary signals?
I had 2 things to say, but now I have 3 things to say. No, in response to your comment, Ed, I'm really intrigued by what sorts of viruses or other microorganisms might now be emerging in melting permafrost. That things that were just not, maybe we should say that we were not accessible to those things, but maybe, we're becoming accessible, and sequencing is something we could do to get some heads up there. For me, if I think about, very large data sets, for me, the issue is not how to build really big trees. For me, the issue is how to manage data sets of that size. So if science is the process of turning data into knowledge, I'm not good at thinking about hundreds of thousands of things at once. So I want tools that can help me take a large data set and pull out representative sets. And so Alexei's talk about how to sample taxons is an example of something that would be really helpful. So, or I want to study something in a particular taxonomic group. I would be delighted if somebody had already figured out not just which genomes are interesting, but which genomes are interesting that also represent not just a broad sample of sequence diversity, but also functional diversity, ecological diversity. So what kinds of tools can we build to help, manage working with large sets? Can we when is it appropriate to subsample? What kind of statistical tests, corrections do we need to make for that? Things like that. And I have a third one if
Go
you
for
wish.
it. Go for it.
So there was a review article that appeared in Tree, I don't know, 2 years ago or something now, called Phylogenetics is the New Genetics. And the basic principle was that evolution is doing experiments, and we can look at convergent evolution and parallel evolution to see how to form hypotheses that could then be tested. And this is something that we can do in species where there are no tools for doing, for example, knockout experiments, or there's no budget for doing, large scale phenotypic studies. And that's something where large trees is something that plays into that. And as a community for building, to thinking computationally and statistically as well as biologically, we can make a big impact.
So I'll bring up one rather ugly issue, I would say, in big trees, and one that I've butted my own head into, and we saw yesterday with ferns, which is the quality of taxonomy. Things have been deposited in GenBank under multiple names in some cases, linking sequences to traceable samples, and especially for macroscopic organisms. With microorganisms, you can have culture collections, although a lot of things are not culturable, so that's a problem. But with macroscopic organisms, we can put animals in various museum accessions and have traceable vouchers. We can do the same thing with plants, fungi, etc. But the linkage between specimen and sequence is surprisingly bad. And this is one I'm not sure if maybe there's a natural language processing sort of solution to trying to look for this information. GenBank does have a voucher field, but it's not required. I've noticed people will put voucher information or something that could be linked to a museum voucher, sometimes in like the definition line or something. Recently I had somebody just simplistically say, okay, go through all the records and just use a regular expression search for voucher, and try to get some information. So I'm not sure how we can move forward there, but that's a challenge.
I think the Phylotastic project made some attempt to use natural language processing to pull out taxonomic information from the literature. And I've tried to use their tools, and it's not easy, but I think in terms of a library of starting places from which one could build, it's an intriguing project.
Okay, so there's one more comment from Janani online. Homogenizing metadata across these diverse data sets is also needed to help with this large-scale integration. We see this a lot with microbial data set too. The microbes also tend to change names every other year.
There's some community of people who wrote papers about C. diff in the 1 year where they had a name that began with a P instead of a C. And I feel so bad for those authors. They went from Clostridium to Peptoclostridium to Clostridioides in 3 years. And the people in the middle.
So maybe as a slight counterpoint also to the earlier view that with viruses that like we almost scratching the surface and we all know almost nothing. In terms of maybe kind of more familiar clades of species, I mean, it seems rather unlikely that we will discover a new phylum of animals. And I mean, there are some parts where we start to have quite a good sampling, and then there's some interesting questions that come up where we can ask ourselves. I mean, will we get a point where we will have characterized all the gene duplication that are altered in a certain time, or at least between 2 times in the past? Will we have characterized all mutations such that we don't learn anything new, and at least from ancestors once we sequence a new genome? I mean, this is in a way a bit what is happening with the COVID data when we are trying to reconstruct like the things that happen. I would say not at the leaves of the tree, but a little bit deeper in the tree. And so does this also give rise to new approaches? This high density. I don't know if you have some thoughts.
Actually, I think it's a follow-up of what Dannie was saying before, that actually we don't know for now, and we don't really want to work with so many data, so many sequences, so that we are not able to reconstruct the trees. But the avenues for this problem are probably the one of sampling, being able to find some sample that are representative of the data set that we have. And usually when we are reconstructing these trees, actually we are interested at different scales. So sometimes you want to look at different groups of species, and then we are first interested in knowing what are the placement of these groups of species regarding each other. And sometimes we are at the lower level where we have a lot of different species as well, for which we want to recompute the phylogeny. So I think it's still possible to always look at smaller sets of species to reconstruct, and then over solutions, over problems that can come with interesting solution are the phylogenetic placement. So when we have already phylogenetic trees, how do we place the new sequence, the new incoming sequences in these trees? So these are also questions that can be addressed to solve the problem of the huge size of data.
Go ahead.
Well, I have a comment on actually how in knowledge that we can acquire with fast short read sequencing or fast inexpensive sequencing that we couldn't before, which is our traditional approach is we have present day genomes or present day proteins, and then we can try to reconstruct. But we're looking at sequences that have experienced both mutation and selection. And so there are all sorts of questions where we might want to understand what the underlying mutation processes are like without the influence of selection. And recently there have been projects where cells are passaged, on a daily basis and sequenced before selection has the opportunity to act. And therefore you can start asking, what are the probabilities of seeing certain kinds of SNPs? And what I'm waiting for, and everybody knows about, if anybody knows about such a data set, what's the length distribution of segments that are gained and lost prior, as a mutational process as opposed to under selection. So I'm looking forward to seeing that data. And if somebody knows of such a data set now, I'd love to hear about it.
Okay, so I think we are running out of time, sadly, because this is at least from here, it's a very exciting discussion. I would like to thank, well, first of all our panelists. But also all the speakers in today and yesterday. I think that was really wonderful. those of you who've contributed also to reviewing abstracts and also all the co-organizers of this session. I mean, it's been, I think, really very exciting to be back in person for an EvolCompGen. And, next year we're going to be in Lyon. So hopefully we can get more of this with also more colleagues and perhaps we'll already have some ideas about solution for some of the problems that we've discussed today.
We can also thank our moderator for the very interesting questions.
And special thanks for Kevin, who has done nothing but solve, boring technical problems for the last 2 days and has, not gotten a chance to enjoy the rest of the conference.
Okay, so there's still a session afterwards, and don't miss the closing. And with the prize announcement, there will be also some prizes for our session in particular.
Right,
and-
and so if you go home, we can't give you your award.
Yes. So see you around. Thank you very much.
Thank you.